All right. Hello. Good evening, everybody. I hope you can hear me all right. Again, I do not have a great internet connection today, so I would not be able to switch on the video. So we'll just have to talk like that. I'll start by sharing my screen, and in the meantime, if there's any question that you have, again, as always, throughout the class, if there's any question, you can unmute yourselves or you can type it in the chat window. And all right. So you should be able to see the screen now. So everybody, welcome to the eighth and the final live session for the NPTEL course Cell Culture Technologies. We have had 7 weeks of course and 7 weeks of these live sessions, problem solving sessions, and this is the final one. So again, before we start, do you have any questions, any general questions or anything about any of the 7 weeks worth of lecture discussed? Or yeah, anything. I'll just stop for a minute, and then if in the meantime, if any of you have any questions, just type it out or just unmute yourselves, and then we'll start with this week's discussion. And I mean, of course, if you have any general comments on the course, on the live session, about the assignments, about the content, any suggestions throughout, or yeah, doubts, whatever it is, either now or towards the end of this class, just let me know. Hall ticket. Oh, I think you should get it like a week, at least a week in advance. So your exams are on the 1st of November, right? So you should get it by then, but then I think it'll be better if you could email the NPTEL people directly, because it would be streamlined for all courses, not just specifically for any course. So I am not in that loop, but yes, you should definitely email the NPTEL team, or if there's any contact number that's there on the website, just talk there. But I think you should get it well in advance. Yeah, okay, great. All right, so if there's nothing else right now, then we'll start the discussion. So, if you remember last week, we transitioned a little bit from neurons and myelinating cells to muscle cells. Okay, we discussed briefly what they are made of, what sort of a differentiation regime needs to be done when you're culturing muscle cells. So today we'll just like eighth week discusses a specific um, it discusses a specific instrument and a technique which is used to muscles and study muscles, and in one specific example that we'll see is also the neuromuscular junction. So I don't think today's class would be very long, but we have been asked to take classes for at least an hour and a half. So I will go a little bit slow in that case, but let's see how this goes, okay? All right, so there are broadly 3 muscle types: skeletal, cardiac, and smooth. So the most focus that we've had is skeletal, a little bit on cardiac, and up until now we've not had any discussion on the smooth muscle, but so we'll continue with that. And there are 2 major key proteins. These are cytoskeletal proteins, actin and myosin. There are different subtypes of each of these. The type of actin, the type of myosin that is used not just in so actin, for example, it's not just present in muscles, but you see it in all cell types. So there's actin filaments and microtubules and these words that you would have heard. So again, different subtypes exist in different cells. In muscle particularly, there's obviously a more deeper role of these 2 proteins. So these 2 proteins, these have chains, they slide over each other, which is contributing to a contraction or an extension movement. And it generates a force. And that is different for each type of muscle. Also, that will be different depending on what age it is. So, especially when you're differentiating it, how mature is the muscle, how much force can it generate, and this changes in several disorders. So, the question comes up, like we had taken the example earlier of some uh, neurological disorders. For example, earlier we had started off with the example of Alzheimer's, where you want to check neurons and neuronal connectivity. Then we also took the example of demyelinating diseases, where the myelin sheath was not being produced either in the central nervous system, which was multiple sclerosis, or in the peripheral nervous system, which was the Guillain-Barre syndrome. So now, when you want to, so now another use case that you would have, like why would somebody culture muscle cells at all, is that when you want to screen some drugs, when you want to quantify what is happening with each drug, or you want to understand some disorders better, then whether it, so depending on what sort of a disorder it is, which muscle is affected in that disorder, you would either grow myotubes or cardiomyocytes in culture. And then one of the major readouts, so in case of neurons, for example, one of the readouts that you would be interested in is how many neurons are growing, how much are they being connected to each other. For the demyelinating diseases, it was the extent of myelination. So that was quantified by whether you have these nodes of Ranvier being made, or just the MBP protein being there, right? So likewise, in case of muscle cells, of course, there's a lot of things you should quantify, whether or not they are dividing enough, whether or not they are differentiating enough. All of that comes. But one of the things that need uh, that require a special apparatus is how much force is being generated, right? You could study. So remember we talked briefly about, or did we? Maybe we did not. Like if you want to talk, I mean, we did not discuss the mechanism, or rather, we did not talk about the technique, but you would want to, if it's possible, you would want to quantify how much impulse, how much electrical impulse is being generated in the neurons, right? How much is the action potential generated, and is it being transmitted long distance? So those have these options where you can insert an electrode in one place, and then you can get a readout of how much electrical conduction is happening there. In case of muscle uh, force generation, it requires a different setup. Okay, so there are these, again, system. We briefly talked about this in, I think, the first or the second week of the course, that there are micro electromechanosystems. So micro is because you're dealing with a micrometer scale, and the system that you'll that is used is mainly to check the electrical, what is happening, and the second thing is what is mechanically happening there. Okay, whether you want to change some mechanics or some electrical things, or you want to record those things. And the main setup is there's some biological components for which you want to study their electronics and their mechanics. Okay, so before we go there, this is a new word that was introduced, which is a cantilever. So there are, so when you talk about bridges or flyovers and all of these structures, also at a smaller scale, like you have balconies at homes. So these are structures which are suspended from a rigid support, but below they don't have any support, right? Balconies, for example, below there's nothing, but they're supported from the side. That sort of a system is called a cantilever. There are, I mean, this is just a screenshot of what are the beams that people use. This longer thing, it's called the beam. So when it's this sort of a setup, the first one, that's called a cantilever. So what applications and why cantilever is used, there are different things. So what would happen is this cantilever, unlike what you see in the balcony. So in the balcony, it's not happening that you walk over it and it's deflecting up and down, right? It's not moving up and down. But if you see the case of a diving board that is at the top of a swimming pool, so if you've seen some videos of the Olympics, or if you've yourself, if you're interested in swimming, so what's going to happen? So what happens is there's a swimming pool and there's an upright ladder that you can go maybe 7-8 steps, and then there's a diving board on which you can jump. So you can jump a couple of times on it, and it keeps going up and down, and then you jump out of it into the pool. So that is a flexible cantilever, unlike a balcony, which is a fixed one. So that's about how much it is reinforced. Anyway, so we will go with the case where the cantilever is flexible. Now, the cantilever's flexibility depends on how thin it is. So if it is thin, it can deflect because depending on how much weight it can take. If it is thicker, its flexibility would be lesser. Okay, so let me know again if at any point there's any confusion or if I'm saying something that doesn't make sense. So, so that's a so that is what is being used. That is what a cantilever is being used for. So, if the cantilever's deflection can be quantified, so you can have like a ground truth that okay, if it deflects, and how do you quantify the deflection? So if I can just maybe draw it out of a small thing, just give me a second. So can you just hold for a second? I'm just muting myself. Hello, yeah, sorry. Okay, so here we were. So if you imagine this horizontal thing is the cantilever, and this is where it gets oh, sorry, not the best drawing. Let me draw it in a different color. So if this is how much it deflects, okay, then there are two ways in which you can quantify it. So one way can be that you quantify this angle theta, or the other way is you can quantify this length, okay, or the height difference. All right. So either way, you now know that, okay, if there's an X amount of force that is placed, which is causing a deflection, it causes a theta angle deflection or L uh, distance or L height or L width of deflection. Okay, so we'll briefly talk about one application where it started. So this is an example of an atomic force, some sort of a topographical measurements. So if you can see here, this is a cantilever. It has this pointed end at one end, pointed thing at one end. So at this end, there's this pointed thing. So what's going to happen is it's going to scan through. You can see here, it's scanning through the surface. So it's going horizontally, left to right, then again right to left to right to left, and so on. And what happens is, so it goes here, and then it gets deflected. So you can see here it's getting deflected, let's say in one direction, some direction. So it's some deflection. And then it goes here, and then there's another deflection at the bottom, and then that continues here. That's what you see here, right? It goes on, there's some deflection, the cantilever continues, there's another deflection, and it continues. So therefore, what it can reconstruct is this topography, that this is what your structure looked like. And this is the length in micrometers. So that's one example of a cantilever, of the application of a cantilever. Now, just so again, these screenshots, you don't need to look at them or understand them in detail. So the the bio cantilever thing, you can really imagine it to look like a swimming pool. So there's this stair, and then there's a diving board that's jutting out of it, and then below it there's a lot of space, for this cantilever to actually come up and down. Okay. I've just attached two screenshots which give you roughly different possible. So of course, there's a lot of different ways of creating that system, and also depending on how complex you want it to be. So you start off with something, you've added some layers, you want to put some extra things on it, and then you say, okay, this is where you start, and then this is etched out. So etching out is a word that's often used with microfabrication. So etching means to carve it out or to cut it out. So basically that place is being removed. And then another things that you want to add and all of those. Likewise here also, it's very, very similar. So all in all, what is happening is that there is a part of this material. Okay, we'll call it the substrate for now. Okay, so that substrate with which the micro cantilever is made, that's protruding out into a space which is empty from below. Okay, so that's the broad setup. And again, cantilever is a general term. Micro cantilever is when your cantilever is micrometers in range. Okay, so now we talked about this example where it was used to get the idea, the topography of a surface. So this is where the bottom part of the cantilever was being used, right? So there's this pin sort of a thing at the end, and that is what is happening. So the deflection, yeah, so that's what, so bottom part is where the deflection is being generated from. Now the other case is, so see, this is a micro cantilever again. You can see that maybe there's some very thin film that's added on it. So we'll come to it, but think of it as this blue thing, the one, the broader thing, which is labeled as the micro cantilever. That's the main substrate, which is generally made of silicon or PDMS, which is polydimethyl siloxane. Just remember these names. This is, so yeah, silica-based components. Because that has the right flexibility, the right range of flexibility that helps in these measurements. And then what you can do is, on top of that, you can, let's say, you can add your, as a thin film. So thin film. So you can add your matrix, your ECM. Okay, for example, if you are growing neurons on it, then you would grow something like laminin. Okay, ornithine, laminin. If you're growing some muscle cells, okay, things, it could be like collagen. Okay, so whatever the substrate, we talked about ECM components a cup until, for a couple of weeks. So whatever you add there, so the thin, so ideally it's that thin film, but it could also be the thin film and whatever is there on the top of it, right? So you have that thin film, and then let's say you have one cell type, you have another cell type, or you have some receptors and then analytes, or you have antibodies and antigens. So whatever this combination is, so this is, you set it up as a baseline that, okay, when I've added just my ECM, I would assume whatever deflection is there, I would call it 0. But now when I add my cells and when they are now contracting, that is when I would say that, okay, that's the deflection that I'm interested in. So we talked about that the thickness of the cantilever is determining how much it can deflect. The thicker it is, the less wobbly it will be, right? So it will deflect to a lesser extent, but again, the ratios will still hold. More cells or more contraction would lead to more deflection and so on. So the deflection angle, so what I drew as theta, that's proportional to the force that is causing that deflection. And for that, it's an equation. Again, it's a complicated equation, so we don't need to go too much into its detail. But yeah, but the Stoney's equation is what relates that stress or that force in that thin film to the curvature that it induces in the substrate. So again, yeah, this is just something extra. So what all would you need for this equation? So the first thing is, of course, you need the thickness of this film. You need the thickness of the substrate. So film is your collagen with without cells. Thickness of the substrate is your silicon or PDMS. Young's modulus of the substrate, so that tells you how much prone it is to being deformed. Poisson's ratio of the substrate, I am not super sure what Poisson's ratio is. If any of you know, then great, but I don't know about this. And then finally, you have the curvature. So finally, what happened? How much did it curve? And then you would be able to calculate. Oh, yeah. So curvature of the substrate is, sorry, it's that how much are you starting off with? Okay. And yeah, so if you're already starting with some deflection and like that, and then finally it's going to tell you that how much was the force that was generated. Yeah. Oh, sorry. The curvature is still yeah, this theta or the amplitude. So amplitude is like this L, that's the amplitude, or it's rather half of the amplitude because you assume it would go as much up as it go down. Okay. It's sort of like that swimming pool diving board. So yeah, sorry. So thickness of the film, thickness of the substrate, uh, another property, some few more properties of the substrate. So that's your cantilever. And finally, how much did that cantilever deflect? So that will finally tell you the stress, which is sort of, we'll just relate it to the force for now, that is being generated by your cells, right? So in theory, this sounds good. Any questions up until now? Oh, we have a lot of people joining us. Hi, everybody. Thanks for joining in. For those of you who might have joined us while the discussion's already started, if you want me to go over something real quick, again, let me know. Otherwise, anyway, the session is being recorded. You can take a look at it later also. But uh, if there's anything you want me to go through or give you a quick recap, let me know. Otherwise, I'll just continue. Yeah, no, all good? Okay. Cool. Let me know again at any point if you want me to discuss something again. So this is the basic principle of how a cantilever would be a good system to measure forces, some sort of a stress that's there on the top. But now, when you're working with cells, there are a few extra things that need to be considered. So if you think about this structure again, so maybe we'll talk about this thing, this point, it's clearer, or maybe even this point. Okay, they are both like good enough things. So what you want here finally is, so this is the place where you want your cells to be, right? So this is where you want your cells. And this point, you do not want any cells, right? You want cells and you don't want cells here because that's not where um that's not where anything is happening, and it, if at all, it can obstruct some measurements. Another thing that needs to be, so yeah, so of course, if the cells need to grow on your cantilever, you have to add the substrate. So, or rather, I should say that you need to add an ECM on that, right? So depending on whatever cell you're growing, so we'll call it the patterned micro cantilever. And then, of course, you need to have space to provide the media, right? So, for example, imagine there will be walls, taller walls here. So, of course, you can fill up this space with media. But again, that should not hamper but then your cantilever is in a media, right? So, think about it. If there's a diving board that's in air versus if there's a diving board that's in water, how much is it going to deflect, right? So there's going to be some extra buoyancy that the media causes, right? So again, these are relatively complicated concepts. So we are not here addressing each of them. And very honestly, this is a super new topic for me also. So I'm just trying to explain this to the best of my ability. If you do have any questions, um, the best thing would be to post it on the Q&A forum. Another thing is you can search for papers that have made up these, which have developed these micro cantilever systems or use them. That would surely help. But yeah, so it's just like a disclaimer. So we are not addressing all questions, but it's good to know what things have to be considered when you're developing a system, right? So the thing is that in that space below, there is going to be media, but you don't want anything growing, not just your cells, but you also don't want any contamination to happen. So, and of course, no leaks or anything from the cantilever at the top. So, cytophobic coatings, literally meaning cyto means cells, phobic means not liking. So, coatings which prevent any cells from growing, like PEG, there are other options also. They can be used to coat the bottom part, the empty space below, so that nothing grows. So now the question is, we said that okay, you want to measure the deflection angle. How do you measure that deflection angle? So think about it, that when your cantilever is perfectly horizontal, you shoot a laser that falls on it, that gets deflected back, and then there is, let's say there's a position sensitive device which has a wide range. But what it finally detects is that okay, it says that it's sort of like reflection. So the angle that it made with the horizontal, that stays the same on both sides. So theta and theta prime are same. But now if you see here, when you have these myotubes that you've finally grown on some ECM and they're contracting and extending, and then there's this growth, and then there's this deflection or this oscillation. So finally you've got a new, let's say a theta. So what you get is now, so let me just draw this again. So this was your original micro cantilever. Oh, wait. Sorry, I'll just use the pen. So yeah, so what this is telling you here is, why is this eraser? Okay, so you have this. So now let's say, I'm not doing a great job of making this. Okay, so this is a horizontal surface. Okay, this is like the absolute horizontal. And then what we have in here is, let's say, this is the sort of deflection that you've got now from the cantilever. Okay, so that's your some theta, or yeah, that's the theta. But how is it measured? So you have this laser that's going on here, and then let's say it goes on here, right? But earlier, when the laser was falling, so again, the laser was still the same earlier. Then it was falling, let's say here. Earlier, so earlier it was this blue laser, and now you've got this, right? So it's a different angle. Again, so that different angle is going to tell us about the deflection that's happening. So rather than measuring this deflection, but again, there are multiple ways in which microcantilevers you can have detectors and these laser sources at different places. So either way, whatever is happening is that you're finally able to measure the angle and see if it's a zero, if the absolute value is zero, or if it's a different value. So you've got that force. You can you've got that angle that can be plugged in with the other values in the Stoney's equation, and you've got the force that is generated by the muscle. Okay, theoretically great, but now we have a problem. So I drew on this, but the problem is that your skeletal muscles, especially, they're not spontaneously contracting and extending. So this force is not going to come on its own. Cardiac muscles, they are spontaneous. They beat, they have this inherent beating in them. But skeletal muscles will only move, they will only contract when a certain signal is given to them. And can you tell me who gives that signal to these skeletal muscles? So when you are moving your arms, your fingers, your legs, any part of your body, when you're moving it, how do you, so of course this is, it's happening inside, but what do you think is making these parts of your body move? Which cell type is causing muscle cells to move? And we talked about them a little bit in the last week, I think. Last week or the week before that? Any ideas? Okay, so it is the motor neurons, as the name suggests. These neurons have the ability. So motor has to do with movement. That's the word's etymology. So these neurons, they're neurons, but their final end. So motor neurons, if you remember from, again, we discussed this over the last 2 weeks. They start off in the ventral horn of the spinal cord, and then that's where they start, and that's one long neuron that's going into a muscle. So if you see, this is that motor neuron. It looks like a normal neuron. There's the cell body. There's nucleus. It has dendrites that's taking the signal. It has axons, and this is a part of the peripheral nervous system. So it has Schwann cells, which are wrapping around the entire axon length. You have empty spaces, which are the nodes of Ranvier. And finally, you have the end of the axon, the terminal, which may or may not be branched. And then finally, it is making a synapse. It's making a junction, to be precise, with a muscle. Okay. And this is a zoomed in version of what is happening there. So again, that is also a synapse. So we have not explicitly talked about synapses. So I'll just take a minute to tell you what happens at synapses. So synapse is also a part of so synapse is made of two components. There's a presynaptic part and a postsynaptic part. So presynapse in this case is, so normally when you're talking about two neurons, so what happens is it's not that the cells are connected. If you imagine this was neuron 1 and neuron 2, then the cells are not connected. There's an empty space, which is called the synaptic cleft. So this is an empty space. And what happens there is, from the first neuron, there are these vesicles, which are lipid bound, sort of empty-ish structures, not empty, but they're not cytoplasm full. So what they do is, these vessels are coming in, they fuse with the membrane. So the material of these membranes, the boundary of these membranes and the boundary of your cell is the same. That's these lipid bilayer. So what can happen is, this can fuse with the membrane, and whatever was inside, it comes outside and it just stays there in this empty space. Now, these are your neurotransmitters. So if you've heard about some neurotransmitters, like GABA and glutamate and acetylcholine and a lot more. So these, so depending on, so again, it's all very well planned. So whatever is released, generally, the receptors for those are present on the other neurons' dendrites. So this is a part of the second neuron's dendrites. So in this example also, what you can see is, instead of that second neuron, what you have now is a muscle. And specifically for motor neurons, what they secrete are acetylcholine. So if you see here inside these synaptic vesicles, you have this thing called ACH. So ACH stands for, A stands for acetyl and CH stands for choline. So this acetylcholine is released in this space, and then you have these receptors for acetylcholine. That means these membrane proteins, they see that, okay, oh, I've got some acetylcholine here, so I need to do something. Okay. So that is what is happening at a neuromuscular junction. So instead of calling it a synapse, when it's a motor neuron and a muscle that are making contact, that's called the NMJ, or the neuromuscular junction. So when we talk, again, coming back to where we were. So what happens is when you're talking about a non-spontaneously contracting muscle, for example, your skeletal muscles, the neurotransmitter which is released, which is secreted at this zone, decides the sort of force that will, so the extent of force that will be generated upon it receives that neurotransmitter input. So what you need to do again, so the story mainly is that your muscle cells by themselves are not going to contract. So you need something else. You need to uh, contract them. You need to give them the signal. So again, it's very similar. It's different, but it's similar to when we said that neurons by itself are not going to be a good model system when you want to study demyelinating disease. You want them to be first myelinated and then talk about disease modeling or drug screening. Likewise, if you want to quantify the force on muscles, the force generated by the muscles, they need to generate that force first. And that can only happen when you have these signals coming in. So you have to culture neurons as well. Now, again, anytime you're culturing, which is called a co-culture, whenever you're culturing more than one type of cell together, it brings problems and the problems stay the same. Are the dividing rates of both the cell types equal? Do you want, what sort of proportions do you want them to be in? Do you need, so what sort of a media would these two cell types grow in? What sort of a substrate? So yeah, so I mean the ECM, what needs, what they need, the media that they need, all of those need to be considered. Another thing is you could also excite or activate, electrically stimulate. Um, so, yeah, so muscle stimulation, I'm not sure how well that works. But then when you say that you want to, you have a neuron and you have the muscle now, both of them together. So how do you ensure that the neuron is also firing actually? So again, those things are there because neurons may not be firing themselves perfectly when they do not have these Schwann cells with them. So do you want to culture a third type of cell? Or do you just want to, put in, like insert an electrode, activate the neuron enough? You don't want it to be perfect, but you want it to be enough and quantifiable and, at least reproducible in terms that you can do the same thing again and again over different setups. So, for example, you have um, muscle and neuron growing without a drug, then you have with some drug, depending on whether your neuron is faulty or whether your muscle is faulty, right? So, when we talk about in terms of drug screening or disease, studying the disease modeling. So, how do you ensure that the neuron is also firing and that firing is finally transmitted to your muscle through this NMJ? So all of these things have to be considered. All right, so what would the final setup of such a system be? So you want two cell types, we're not going into the third cell type. So you want your myocytes and you want your motor neurons, okay? Again, remember motor neurons are a part of the peripheral nervous system. So there's one long motor neuron everywhere. They start from the ventral horn, which is a part of the spinal cord. And we saw this, that the best time to take this would be E14, and it barely has any glial contamination. So you're sure that at least glial cells are not going to overtake. And then you want myocytes. And again, it has been optimized that E18 is a good time point. Now, of course, in case of, so it's a little older mice, and also these myocytes, you will have fibroblast contamination. So we have read about this earlier. You do want a relatively pure culture because fibroblasts, like glia, they're going to overtake. So you do want to isolate or you do want to purify your myocytes. And we've seen different things, right? So if you remember, we talked about density centrifugation, right? We talked about immunopanning. So immunopanning or differential settling. So differential settling, we have not explicitly called as a technique, but it's just that one cell type sets first and the other cell type doesn't. So that's that. Or you can have your other methods that we have seen earlier. So that is your Gradient centrifugation, not gravity. Gradient centrifugation, you have immunopanning, and the third thing was FACS, right? So the way people have optimized this is you first do your myocytes, pure myocytes, grow them for a few hours, and then you add neurons. And then your NMJ, so of course it's going to take time for your neurons, for your motor neurons to find these muscles and make good, strong NMJs, neuromuscular junctions, that forms in 15 days. And then what you can do is you can see the electric. So what you do is you take microelectrodes, again, electrodes, which are small in size, which are on the micrometer scale. So you stimulate the neuron and see the activity in the myotube, and that is going to be quantified. So not the electrical activity, but then it will consequently generate your contractile force, which can be quantified by your microcantilever system. So what needs to be done is that your motor neurons have to be stimulated through this microelectrode. So again, you have your microcantilever, on that you have your ECM, on that you have motor neurons, and myocytes growing. And then what you do is, when you think that you have good NMJs formed, then you take your microelectrode to, you direct it to a motor neuron, stimulate it, and then you'll observe that there is contraction, as a result of which there is a deflection in your microcantilever. So your theta is calculated, theta is determined, and then that stress can be quantified. Now that's one example. Now what you can do is, now you can add some extra agents. So what you can add is, of course, we talked about that this setup can be done when you have good myocytes and when you have some bad myocytes. Okay, so on diseased myocytes. And then you can simply compare if you have the same number and everything, is the generated force lower or higher? Another case you can do is, if you have bad myocytes and you add some drugs which you think can improve your myocyte ability or division or something. The third thing can be that you take good myocytes, good everything, and then you add some blocking agents. Now, we briefly talked about this acetylcholine and acetylcholine receptor. Okay, so the receptor is a protein. Acetylcholine comes onto it. Receptor sees it, it's there, and it does something. Now, typically, what that something is. So I'm generalizing this, but this will be applicable in. Oopsies, sorry. This will be applicable in most cases. Again, so if I draw that this is your lipid bilayer, okay? And then let's say here is where you have your receptor. And let's say this is your extracellular space. And this is your, this is your cytoplasm. So what we are mainly looking at is, if this was your cell, right? This is your nucleus. And of course, this is like a bilayer, right? Cell has lipid bilayer. So this is the area that we are looking at. Oops. Give me a second. So this part is what we are looking in here in this figure. Okay. So what is happening now is, so this is your receptor. There's a part of the receptor which is, let's say, inside. I'm just drawing it like this. And then there's a part of the receptor which can fit in your acetylcholine. What color should I use? Blue. Okay. So let's say there's a lot of acetylcholine that's here. So this acetylcholine can sit here. This is ACH. Now what happens is, when this acetylcholine sits on the receptor, or for that matter, any ligand sits on its respective receptor, the conformation here changes. Something changes here. So conformation changes. And then once that changes, then there's a downstream cascade of signal that goes on that finally says, okay, something is happening. Now, when there is a blocking agent, so two names were discussed, bungarotoxin and curare. What it does is they look very similar to acetylcholine. Okay, they can bind to the receptor, but they do not introduce these conformational changes. So, acetylcholine, it binds, and there is a change in the cytoplasmic or cytosolic um, part of the receptor. But when you have, let's say, curare, it also binds, but no change. So now imagine you have a lot of acetylcholine is obviously there because there's a neuron that's coming and sitting on the muscle. But there's also these extra things that are here. Now those extra things will have a competition. So let's say you have 100 molecules of acetylcholine and you have 100 molecules of your toxin. Some receptors may bind to something like that, but not in all of them that you will not have these conformational changes. Or if your toxin is if you have a lot of that toxin, then there's absolutely no chance that your acetylcholine will have space. So toxin is not going to bind here at all. There'll be no conformational changes at all. So your muscle is finally not working, right? So that's why these are called neuromuscular blocking agents. They act as antagonists of the acetylcholine receptor at the neuromuscular junction. So this is a relatively complex concept, but this is how mostly in a lot of places, this is how antagonists work. So they can bind and prevent the binding of something else. And when they bind, they will not let the second step happen, right? Okay. If there are any questions about this, let me know. Because then we just finally have like one more slide, which is just an extension of what we've talked about until now. No questions? All clear? Okay, good. Again, but yeah, if you have questions, let me know. So we talked about neurons and all until now, but we never talked about what sort of neurons they are. Okay, so we said, okay, motor neurons and hippocampal neurons and all of those. So briefly, so again, there are a lot of neurotransmitters. You have glutamate. So we'll talk about these three for now. So glutamate, GABA, and acetylcholine. And depending on what it is that the neuron finally works on, you can either have glutamatergic neurons, you can have GABAergic neurons, or you can have cholinergic neurons. Okay. So if you want your neurons, when you're talking about these E18 and E14 neurons, what you do, what people have done, again, this is like a ready setup. A very small amount of glutamate is added to them at the time of when they are being plated. So again, we something I missed here is, so you have this synaptic cleft, and there's a lot of these neurotransmitters that are released here. Now, not everything is going to be used up by the next cell. There would still be things that are just floating around. Now, that thing is where astrocytes or your PNS complement, which is the satellite cells, they come into the picture. They're also all glial cells. Now, this is what those are what clean up this extra neurotransmitter. So once it's released, it'll be there for some time. It does what its job, and then they need to be cleared. You don't want that signal to just keep happening. You don't want the neuron to keep firing consistently. This is where disorders come into the play, right? If there are diseases where you have too little astrocytes, then this cleaning up will be messy. But you also don't have astrocytes when you have this pure culture of neurons or when you are just focusing on Schwann cells. So if you add glutamate, initially it's good because then some of your neurons can, they'll see glutamate, maybe there's some sort of a feedback loop that, so neurons initially will have these glutamate receptors, but then there's like this positive feedback loop that's introduced, which says, okay, since you see glutamate, you need glutamate receptors. But you don't want too much of that glutamate because there are no astrocytes, that glial cell, to clear up that extra glutamate. So if you still have glutamate there staying for a long time, this is an excitatory neurotransmitter. So what it causes hyperexcitability of neurons. Okay, so in the previous class, I said that neurons can be regenerated, but my question is how this get the signals which were transmitted to the old neuron when they replace the new ones. So neurons initially, when we talk about embryonic neurons, that is where they have some dividing capability. So regeneration, so there are two things actually. One is when the neurons are being divided. So you have one neuron and now it divides to give you two neurons. The other thing is how long or where are they making these synapses? So just the dendrons and their axons, dendrites and axons are moving in different directions. So that can happen. They can make contacts, they can change these contacts, and that is what is happening in an adult brain even now. When you say different memories are being made, that is what is all is happening. Your neurons are making new synapses all the time. But but division is only happening in your younger neurons, embryonic neurons, and not the adult neurons. But so what you're asking is, so yeah, so the way, I mean, that's a good question. So initially, when the neurons are dividing, they would not have any of these contacts. So whenever there's a new neuron that's dividing, again, you have to think of it that these specialized neurons, the complexity of neurons that you see, when they are really complex, when they have all of these specialized axons and dendrites being made, at that point, they don't really divide. When they're still like smaller, not very contact, when they're not making a lot of these synapses, that's when they divide. But then once they have established these contacts, then mostly they don't divide. But that's a really good question, actually. So thanks for asking. And did I answer your question? Yeah? Okay. All right. Thank you. Really good question. All right. So, yeah. So when you have these neurons, you can add some. So let's say you add glutamate. That's an excitatory neurotransmitter. So it ensures it does increase their excitability. When glial cells are added, so like we talked about Schwann cells and all, so it gets better. People have seen that there is a higher expression of ion channels. But of course, you can see that you have to ensure that, again, glial cells are not growing too much. And then finally, so this is your early embryonic neuron cultures. Then finally, when you have an adult neuron culture, so what needs to be done is there needs to be some sort of a temporal conditioning that needs to be done so that they can regain their electrical activity. So something on the lines that you asked. So if you're taking adult neurons and then you're plating them, you initially made a single cell suspension, right? So they've lost all their contacts. Now you need to give them a lot of time to stabilize. So which is why this culturing it's a long process. You can see it needs something around 30 days for them to stabilize. Finally see that, okay, that's there. And then you feed them So again, optimized protocols, glutamate and serotonin is fed for another 30 days so that they can have their receptors. So again, some time for glutamate, some time for serotonin, some time for acetylcholine, and then they can make contacts in the meantime. They can express some receptors in the meantime. And then finally, uh, at, after at least something around 60 to 75 days, they'll be in a good place for you to start evaluating their electrical activity. Okay. But yeah, so you can see that so again, it sort of comes up from the question you asked. We're not exactly, it's not about regeneration or old and new neuron in this case, but still your question holds that they've lost their contacts, right? So the initial signal transmission was not happening. So you need to give it time. You need to give the neurons time for them to make newer contacts. All right. So I think that's about it. That's everything that was discussed this week. I have some practice questions for you, which will just help you in your assignment. And from what I've seen in the past years, also, the exam questions would be very similar in their pattern to what you see in assignments. So they're all going to be MCQs, and yeah, one correct option, and they'll obviously be within the things that were discussed in the lectures. So if you've seen all the recorded lectures, um, that should be good. And of course, attending these live sessions or watching the recordings of live sessions would be a bonus. But yeah, okay. So I have some practice questions. Again, as always, it'll be great if you could be more interactive in these questions. Otherwise, it's okay. I'll let you know the answers anyway. All right. So what's the main principle behind a microcantilever that is used to study muscle contraction? So just say A, B, C, D, and it's okay if you're wrong, no problem. All right, so we have B here, so that's correct. We talked about that this is a mechanical force which causes the deflection. Neuromuscular junction is made at the junction of what? Is it muscle and motor neuron? Is it glia and motor neuron? Is it hippocampal neuron and osteoblasts? Or is it none of the above? Yep. So that's muscle and motor neuron. Would any other people also like to try? But otherwise, it's okay. You can continue answering. So, which units of muscle cannot be used for force measurements? So this is a slightly, it's not a tricky question. Just think about it. So do you remember the order in which these muscle cells divide and differentiate? Anybody? So what's the first thing that you start off with? So the first that you start off are myoblasts. They are sort of like your stem cells. So these have not yet fused. So you start off with your myoblasts. For now, we'll just call them, they're like myocytes. And then you have myoblasts. And then what you have are myotubes. And then you have the final myofiber. Okay. So this and this, they do generate force, but your myoblasts and myocytes, they would not. So your answer here is going to be myoblast. All right. Okay. Right. Which microfabrication technique is most commonly used to produce the silicon cantilever? I think I did not explicitly talk about this, but if you want to give it a try, if you just want to give it a guess, what do you think sounds the most possible? Yeah, so that's photolithography. So lithography is when you are constructing something, some surface thing, and photo is, of course, when you're using some lights for it. Again, I have very little idea about this, but so mainly why it's called photolithography is if you see here, you see that there are some masks and all that are used, right? So what happens is on top of this, you put some mask, which is like it's like the opposite of the structure that you want. And then finally so it's like this, like it's a photoresist, and then you put it on some places, and then everything which, so it could be UV or something else. So wherever you did not have that extra layer is where your first thing is going to be removed from. So you get these grooves and all of those structures like that. But yeah, I maybe I'm not doing a great job at explaining this. So it's photolithography, and etching is, we just said that it can be there to remove whatever parts you want to be removed so that that empty cavity structure can be formed. Okay. And then Bungaro toxin, it causes paralysis. So that's where these neurotoxins or neuromuscular toxins, they cause some sort of a paralysis. And yes, so Bungaro toxin does it because it blocks the acetylcholine receptor. Then when a microcantilever is used to detect a neuromuscular junction, the muscle cell is typically co-cultured with, that's easy, right? So you don't need astrocytes, you don't need clearing up as such, you don't need Schwann cells or oligodendrocytes because your main idea is not to myelinate something, but what you want are motor neurons. Then a functional neuromuscular junction can be confirmed by what? So what do you do if you want to, you've formed these things. So we discussed that, okay, fine, you can make a neuromuscular junction. Let's say it takes you 15 days or 30 days for your motor neurons and your muscle cells to sit up and make these junctions. So you can check that, okay, acetylcholine is being released. So because of course some acetylcholine will come up into the media also. And then of course muscle contraction is the main star of this week that we have been discussing. And I think this is the last question. So in an integrated lab on chip model, so lab on chip is mainly that there'll be very few things you would have to do outside. So everything is going to be in that setup. So we talked about that the microcantilever system, where you have your motor neurons and you have the muscles, you would need one extra thing to start that, to see that whether the force is being generated. What was that extra thing? Yes, right. So micro electrode arrays, or we'll just, so array is mainly when you have a lot of something. So micro electrodes are like that. So yeah, so I'll just show you a setup of the micro electrode array in a second. So this is where you have a lot of electrodes, and because you don't want to just, I mean, it's possible that you can have one electrode and then you're stimulating something, but then uh, you can also have an array where all the neurons are being excited, and then you can, so that finally a lot of things can be done together, a lot of measurements can be done together. So that's where your microelectrode arrays come into the picture. All right. So that's that. So I will just quickly Google something here. So we'll have the microelectrode array. So I mean, of course, you can Google this up, but so it looks something like this, right? So you have a lot of these electrodes here. And for example, you can see here that you have this well sort of a shape. So at the base of the well, you have these electrodes, and then on top of those, you can grow your neurons, for example. So something like this, some incorporation of this sort is what can be done even for a microcantilever setup. Okay. Now, the last thing that I want to do today would be just quickly discussing last week's assignment. So, which techniques allows sorting of cells based on fluorescent labeling? That was facts. What is required before using the density gradient centrifugation for cell separation? So, of course, you need single cells. You need to have a single cell suspension. When would or why might the density gradient fail? That would fail if your cells have similar densities, and that's why you have FACS or immuno-based things. So which marker type is crucial for immunopanning and FACS? So that's a surface marker. But also for FACS, we discussed that if suppose an internal protein can express a color or a, so for example, a lot of times what people do is you can have a GFP tag on some of proteins that are inside the cell, then intrinsically your cell is going to glow green or any other color. And that can also be used. But yes, typically what is done is because you want to label something with antibodies from the, so your antibodies are not generally going to go inside your cell. You don't want to, break up your cells in the process. So you would prefer that antibodies, which would have this color, this fluorescent marker, they can detect something that's on the surface. So surface markers are what I used. What's a critical requirement for cultured neurons to be considered functional? So they have to fire action potentials. What structure is often used in adults to obtain satellite cells for skeletal muscle culture? So this was not, I don't think we explicitly talked about this, but skeletal muscles are, oh yeah, sorry. This is not the astrocytes satellite cells. This is your myoblasts, right? So those satellite cells, yes, we talked. This was hind limb, for example, the rat P1 to P5, P4 hind limb. Sorry. Yeah, and then what is one role of satellite cells in muscle culture? That they have to become yours. They are the stem cells that will finally differentiate into myotubes and then fibers. Next up, which muscle type needs to contract when cultured to prove its functionality? So spontaneously, it has to be the cardiac muscle cells, but ideally also your skeletal muscles need to contract. So you can see that question number 8 and 9, they both need to contract ultimately, but cardiac muscles, they have to contract, and skeletal muscles have the ability to contract, but they can't spontaneously do unless you have a neuron which is stimulating them. And again, another question about FACS is, why is it important to know the developmental timing? Because not all surface markers will be expressed at all times in a particular cell type. So surface markers can change during development, and that can affect how precisely you're sorting your cells. So that's your assignment 7 solutions. And then that brings us to an end of today's lecture, as well as to the final live session of this course. We would not have another session to discuss the assignment solutions of this week. But again, you have that Q&A forum. So if there's any questions once you get the solutions posted on that. And that's about it. So your assignment deadline is day after tomorrow. Today's recording and the PPT will be uploaded to YouTube and the Google Drive respectively. And that's it. So if there's nothing else, then I would stop sharing my screen. All right then. So thank you everybody for joining in, for joining one or more sessions throughout this 8-week course. It was a lot of good learning for me also. A lot of topics were new. I had a good time preparing for these classes. And I hope these live sessions helped you a little bit in understanding the course better. Thanks for joining. Thanks for asking questions. And good luck to all of you, for whoever's registered for the exam. And regenerative medicine. So, all right. So would you like to elaborate, like, would you want a course, or what are you exactly looking for? So, like, I mean, so courses and IITs. I am not sure if there are courses which are floated by NPTEL. Oh, MTech courses. Hmm. Hmm. I mean, I am not particularly aware if there's a dedicated course. Um, I think the best way to go about this would be to actually check the curriculum, the courses that are offered. So, and of course, sometimes it happens that these courses, like it may not be like the entire, like, okay, so two things actually. So, one is if you want to understand the theory versus if you want to also have some practical lab work. So, for example, in IIT Delhi, there are labs which are working with um, some cell lines. There are labs which have some biomedical background. So, because there's a biomedical department, there's a biotech department, there's a pure biological sciences department here. Um, so, so of course, there's like in addition to MTech, there's also this MSR program, which is Masters in Science with Research. So you do have to do some courses, but also you can do a year long or more in a lab. So so depending on if there's a lab in that particular IIT or any other institute for that matter, then that's one way to go about it. Also, depending on if people are working, so regenerative medicine, typically you will start off with stem cells, right? So depending if there are any stem cell labs, which, maybe not masters, but they could be some PhD courses, and then there might be some coursework also associated in that topic. Um, that can be done, but I think you'll just have to search about it. I am not exactly aware of it directly. Um, I would still share my, yeah, if you want, I can just share my uh, email and then just think about it, like read and just try to find if there's anything. Um, maybe you can like just email me and I can help you with, if there's anything I can help you with, then surely that can work. Um, so like my work is also on working with stem cells to make um, neurons and brain organoids. But mine is a joint PhD with the University of Queensland in Australia. So I did all of my lab work there. Here at IIT, it was mostly once these samples were made, once we had the final things made, these were sequenced, and the analysis of all of that is what I'm doing here. So I'm not aware of which labs do that. But of course, there are a lot of things that are coming up. So I would be inclined to think that most of the newer labs would be of your interest. But yeah, but also I would recommend you not to limit yourself to IITs. So if you've already done a BTech, then you may, and maybe a year or two of experience somewhere, then you may be eligible to directly apply for a PhD. And there are institutes like NCBS, TIFR, um, yeah, ICERS, NICER, all of these places where newer things are happening. So definitely explore those as well. And yeah, in some of these, there are master's courses available. So yeah, so you can think about it. And if nothing else, you can for sure do some internships if practical experience is what you are more interested in than coursework. Also, keep an eye on the next semester's NPTEL list. So mostly every alternate semester, the courses are similar, but they could be changing. So see if any of those are of your interest. Yeah. Okay. Sure. Check IISc, of course. Yep. All right. Great. Thank you so much again, everybody, for joining in. Good luck with your exam. Good luck with the other NPTEL courses if you have any. Good luck with your studies, and thanks for joining in. So bye-bye then. I'll end the meeting. Take care. Good night.